My lab is really interested in influenza pandemics and how they arise. We know that all influenza viruses start off in wild birds. There's a huge reservoir of different subtypes of flu there. And occasionally, those viruses manage to cross the species barrier and infect us and cause pandemics. But luckily, most of the time, the bird viruses don't infect humans very well because they bind to the wrong receptors or because their polymerases don't work well in human cells. Occasionally, avian flu viruses can find themselves replicating in pigs, and in pigs we think that those viruses can become adapted so that they can better infect humans, or they can even undergo reassortment, which means that they can cross the species barrier better. Viruses from ducks can also infect chickens, and there's a lot of press worry about chicken viruses infecting people and causing the next pandemic. But actually, what we try and put forward in our article is that in chickens, the virus undergoes adaptations which may make it less likely to infect humans rather than more likely. And we describe those adaptations as glycosylation, the changing of the neuraminidase gene, and other changes in haemagglutinin, which I'll explain now. So we know that influenza virus, when it wants to infect humans, has to negotiate a special type of innate barrier, which is the mucus layer, which overlays the apical surface of the human respiratory epithelium. And what we've shown recently is that avian viruses that have replicated in chickens have truncated the neuraminidase stalk, and we think that that makes it less able to penetrate the mucus barrier. Whereas with long-stalk neuraminidase, the virus can cut all the way through the mucus barrier to the apical cells below and initiate infection of the respiratory epithelium. So viruses that have been in chickens with the short-stalk neuraminidase get stuck in the mucus, and therefore they don't cause pandemics in humans. Another feature that happens when viruses cross from ducks into chickens is that we see a difference in the stability of the HA protein. Now, why does this matter for transmission? Well, because if we consider haemagglutinin, it is a naturally unstable protein at low pH. Influenza virus enters cells through the endosomal pathway. And as the virus passes through the endosomes and experiences a decreasing pH, it eventually gets to a pH at which its HA undergoes a conformational change and fuses the viral membrane with the endosomal membrane, and from there the viral genome can be released. Now, we and others have made measurements of the pH at which this happens for chicken-adapted viruses versus human viruses. And if you look at the red viruses in this diagram, what we see is that they undergo this conformational change at a relatively high pH. In other words, they can fuse in the early endosomes. This is good because it means that they release their genome early during entry and get going with replication fast, and also possibly because they might avoid restriction factors like IFIT-M3, which sit in the late endosomes and might otherwise stop the virus from infecting. On the other hand, we see that when we make measurements of the human HA, its HA takes a lot longer to fuse. In other words, it requires a lower pH to undergo that conformational change and release its genome. This means that sometimes it's susceptible to the restriction factor IFIT-M3. But overall in the environment, what it means is that the viruses are more stable. And when human viruses are trying to pass from one person to another and experiencing the mildly acidic environment of the human nasal mucosa, they may need that extra bit of stability in order to survive in that environment. What we hypothesize is that in the poultry transmission environment, the viruses don't have to survive as long, and therefore they can afford to be relatively unstable. Adding all of these together, what we hypothesize is that chicken-adapted viruses are much less able to successfully transmit through the human transmission environment, and therefore the next pandemic may well not come from a chicken-adapted virus at all, but more likely from wild bird sources or from pigs.